fireside chat tonight.
Great. Yeah, thanks for Mark. Thanks for having me. It's nice to be able to virtually chat with everybody. So yeah, so you can see here, here's a picture, and somebody had asked about wrapping or unwrapping, and I have some info at the end that we can talk about. And I presented on that before, and I talked about it a little bit at the Illinois State meeting last winter, and we had run a project over the winter before last, actually, looking at hive wrappings in the central Illinois climate, where a lot of recommendations argue you don't need them. But I've been really interested in winter bees and wintering bees for a while. And so I wanted to talk about something a little bit different that we've been doing in my lab to look at susceptibility of bees to diseases during the winter, which is something we really don't know that much about. And kind of some new projects. And so one thing I will warn you, this presentation is not super polished. A lot of this is really new stuff that we just did in the last year. And so it's still things that we're, figuring out the best way to present. And so, like I said, I'm really happy to entertain questions and to hear feedback from people and to hear what you guys are seeing in your own hives. Because a lot of the reason that we did this project was based on observations that I had overwintering colonies in Iowa, where I worked at Iowa State University for 5 years before I moved it back to Illinois, and then overwintering colonies here. The first thing I want to do is point out all the other people that worked on this. So some of you might have even seen some of these people present at other meetings. So Ashley St. Clair, she just presented at one of the St. Louis beekeeping meetings recently. She did a lot of this work. She's a great beekeeper. Alex Payne just joined my lab from Texas A&M, and she's done a lot of bee school stuff down in the Southwest and the Southeast. Nathan Beach, some of you might have met. He's the manager for our bee research facility. And then Gyan Harwood is a biologist who just left my lab and now works for Corteva Agrisciences. And so they all worked on this project. So this is a project. It's one of these funny things where it was nobody's really job to work on it, but I kind of forced everybody to work on it a little bit. And so over the course of the years, lots of people had a little bit of a hand in this work. And so if you're interested in any of the other stuff we do, we have a website and you can kind of check it out. And it generally is up to date. And if you're ever interested in some of the research we do, if you go to the publication section, I always post any of the publications or any of our research on there. Some of it, if you just find it on the internet, it'll ask you to pay for it. It'll be like $35 if you don't have a subscription. But if you go to the website, I just illegally post it on there. And just you can just download it from right there if you're ever interested. So what got me interested in winter? So of course, wintering biology and honeybees is really cool. Temperate winters like we have in Illinois are hallmarked both by cold temperatures, but an end of forage, right? There's no more food. And so you have this whole mixture of things going on inside the colony. And one thing that makes honeybees so special compared to the vast majority of other insects is that they don't hibernate. Most insects go into diapause and they go to sleep and hibernate for the winter, but honeybees, they remain really hot and awake the entire time. And so how do they do that? And there are ways they do it through this process called winter bees that I'll talk about in a minute.
You can watch it, or I can bring it to your lab.
No.
It's just don't.
And then at the same time, we know, we at least we think, usually that winter is considered the highest risk period for colony survival, right? That people lose your colonies during the winter. Winters are the time it's the hardest. And we think about this as being the time where colonies die. There's no food out there. They're trapped inside the box. It's really cold outside. And we know that colony losses are high amongst many types of beekeepers, although this is from the Bee Informed Partnership, which I'm not going to comment on some of the issues that we have with the way they present data, but people lose a lot of colonies, especially in the Midwest, and especially during the winter. And here in Illinois and states like Iowa, where I first started working on this stuff, they're reasonably high all the time, year to year. And when we survey beekeepers, we don't really have good historical data for colony losses, but beekeepers generally say, I'm losing too many colonies. Colony losses over the winter or over the year are too high. higher than we think is appropriate, higher than we think we should be losing. And these are particularly bad amongst backyard beekeepers and hobby beekeepers. As we know, beekeeping is valued in the millions of dollars, and it's really valuable for pollination services. And so bee losses are costly. They have a big cost. And a lot of these bee colonies are in some part of the year, at least, located in this region here that is in this kind of agricultural ecosystem that we work in. So that's what we're interested in it. So when we ask beekeepers, or you go read beekeeping books, and here's a beekeeping book that I really like, and this is the one I used when I taught beekeeping courses. What do you need to do to have bees go into the winter healthy? Well, we always say we've got to control varroa mites. There's lots of ways to do it, but if your varroa mites levels are high, that's generally bad. You need to have a colony that's queen right. If the queen is dead, or the queen is very old, or the queen has other problems, your chances are of coming through that end, even if the bees are of good health, coming out the end where they start to build back up in the spring, very unlikely, right? They've got to have adequate food stores. And so this means that depending on your climate and your conditions, the bees just have to have enough food to eat and produce the heat that they need to get through the winter. And they're not hibernating, so they actually need a lot of food. And that's going to differ on how cold your winter is, when your first blooms are, and when your last blooms are. And then there's a lot of recommendations, some dating back 100 plus years, on using different types of protective environments, ways that as a beekeeper, we can control the hive, control the nest. This can be things that we think of as pretty simple, like entrance reducers to prevent robbing behavior, entrance reducers to help them keep the colony a little bit warm. It can be things like moisture boards or top entrances to help control moisture buildup. As a bit of a side story here, I first learned how to keep bees in central Arizona. And so when I first moved back to the Midwest, I lost almost all my colonies the first year because I had never thought about moisture control. To me, moisture control was pouring water into the colony. It was making sure you had more water available, because I was used to beekeeping in 120-degree weather in the desert. And so moving to the Midwest was like a huge change for me. And then, of course, things like hive wrapping. And then, as I can talk a little bit about later, moving colonies into buildings, which has become really popular in some circles, especially out west. And so there's this long history of beekeepers doing lots of different things, lots of different management strategies to help bees overwinter, especially these protective environments. And I can get back to this a little bit later, but again, if you go back 100 years to beekeeping books from the early 20th century, those recommendations would tell you, if you live in northern Illinois, don't even try to overwinter bees outside. It's a waste of time. Put them in a building, put them in a cellar, dig a trench and put them in a trench. And if you live in more lower climates, you've got to wrap them, and they've got to be wrapped in a bunch of different things that people use then to wrap. And then only if you live south of basically, probably south of like where like Staunton is, and that farther down, that's the only place you should be trying to keep bees outside. We obviously know that's not true. But people were doing that kind of thing even 100 years ago. But clearly bees overwinter outside. They have overwintered without beekeeper interventions. They can overwinter under lots of different conditions across the world. Cold conditions, warm conditions. There's even, bees live places where there is no winter, but there are long dearths in forage. For example, there's evidence for winter bee conditions occurring in hot climates in the middle of the summer. Again, when I was doing the first bee work I ever did in Arizona, we actually shut down all of our research in June and July and part of August because they stopped rearing brood during that time. That was functionally our winter. There was little food to bring in. There was very little brood in the colony. It was just too hot. You had to protect the colonies, because if you didn't, small colonies wouldn't freeze, but all the wax would melt and it would come out the front of the colony and they'd all die. But bees are flexible and they can deal with this. So what are the bees doing? And so the biggest way that bees deal with these stresses is through the production of winter bees. And winter bees break all the rules. They do everything different than what we think about the bees that we see day in and day out in the summertime. So in the summer, we've got bees, they've got short lifespan. We usually think of bees, worker bees living about 4 weeks. They come out as a day-old newly emerged bee, clean a little bit, they become a nurse bee, maybe a soldier, then they go become a forager, then they die. But winter bees, they don't do that, right? There's no leaving the colony. It's winter, it's snowy. Sometimes the colonies might be buried in 4 feet of snow. And so what do those bees do? Well, they just stay in there and they live for a really long time, right? The queen isn't laying any eggs. She's not producing new brood. So those bees have to live a really long time, 5, 6 months sometimes. Summer bees, people have been using bees to study learning behavior for a long time. Just like Pavlov's dogs, where you could train a dog to drool at the sound of a bell. You can train bees to stick out their tongue at a smell that you've trained them to. And people have used this to study learning behavior and kind of how neurons work for learning. And summertime bees go through a very short term, get smart and then get dumb as they get older. But winter bees, this whole long lifespan, remain smart. They can kind of pick it up and kind of go at the end of the season or the end of the winter. Summertime bees emerge, they eat a bunch of pollen, they're kind of fat, they put on a lot of fat, but by the time they become foragers, they've lost all their fat and they're very thin, very skinny, not a lot of fat stores. Winter bees, they stay fat this whole duration. And then, like I said, summer bees, they go from being, hatching to being a nurse bee and taking care of the larvae. And they have HPGs that I've abbreviated here, hypopharyngeal glands, the glands that make the royal jelly. And they have really big, juicy ones when they're young, and then they get small and dinky looking as they get old. And winter bees have weird hypopharyngeal glands. They're really big, but they're not doing anything. They're just kind of there waiting. So the moral story here is that winter bees are basically these fat, kind of smart bees just waiting, and they're just waiting for the summer to come. And so they're not transitioning to becoming foragers because there's not foraging work to do. So they're just in there, just hanging out. And so again, as an aside, that it's not really important for this story, people have been studying these for a long time to understand how they do it, because you basically have the same genetics, same animal, but in different environments can live 10 times longer and remain smart the whole time. How do they do it? And so here we can even see an example. So on the top left, this is the inside of a worker's abdomen. We can see on the left side, this is a bee who is a forager. She's used up all her fat. She's not, she's thin now. And on the right, we have a fat bee. You can just see there's a lot of fat inside that bee's abdomen, and we can measure that very easily. On the bottom here, we have the hypopharyngeal glands. You see they look like a little bunch of grapes. On the bottom, we see that when a bee emerges, hatches out, you see that little one crawling out of the cell. That's what those glands look like. They're not developed. They don't do anything. That's why a day-old bee can't nurse larvae. They can't do a lot. They don't have the glands for it. And so they have to eat a lot of pollen and mature. And then we see by day 9, those glands have gotten big and juicy and they're white, and they can make royal jelly very well during that time. And then later on, as they get old, those glands will deteriorate again. It's just their natural life cycle in the summertime. And here's a figure from a paper. This is kind of just like a boring graph, but you can see how it shows that a bee, starting at day 0, its gland size about this big, and then it goes up and it's pretty big on days 6 to 9. And then as it gets older, it declines until, this is a bee that's 33 days old and it's got very small hypopharyngeal glands. And here we see a W, this winter bee, kind of in the middle, has maintained these like kind of big glands the whole time, but the glands don't do anything. They're just sitting there. And so how long do these bees live, and when do they make them? So we think of winter bees, you think, okay, I'm going to crack open a colony in December, probably have winter bees, right? But when do they start making the winter bees? Well, remember, it takes about 22, 23 days for an egg to get laid, develop, pupate, and come out as an adult. And so if queens are stopping laying eggs in September and October, that means that to produce the winter bees, to fill your colony, you really got to start early. And there was some really, I can't imagine, painstaking work that was done about 20 years ago by a researcher named Heather Madela, where what they did was they took a big group of bees, they marked them all with paint, and then they dumped them all into colonies, 4 different colonies. And every 2 weeks, they went through those colonies and they counted how many were in there. And so you can see I've zoomed in on group D, just one of the colonies here. And you can see if the group that was added on the middle of July, by the beginning of October, 100% of those bees was dead. Beginning of August, 100% were dead. And so you can see here, all these bees added that were produced basically in the middle of the summer were all dying and dead by November 1st. But then we see this switch in their study about October 18th, where bees that were added here, they all started to die, but then at this switch point, the curve like flattens. And bees that were added after this point survived November, December, January, February, March, April, late April. And then it wasn't until late May and even June that some of those bees had died. So here we see that these bees are getting produced in this colony mid-October. And if you zoom in on group A, it actually happens even earlier. And so they calculate this out to show how many winter bees are being produced in this colony. And we see that it kind of goes, over time, starting as early as September. And so to me, that's really important to know, because when you start to see problems in your colonies, and we often see problems in our colonies in August and September, it underlines how critical of a time that is, because not only are those your end of summer bees, that's right when your colony is producing these first generations of winter bees. And these are bees that not just have to live and forage and make honey. These are bees that have to see your colonies through till June sometimes, which is just mind-blowing. One caveat I will say here, though, is all this work was done in Ontario, Canada. So the dates are almost certainly shifted a bit for us, but the point still stands that I think winter bees are being made sometimes much earlier than we might expect. That's so weird. So when you think about all this stuff that you're doing, you're controlling mites, you're wanting a good quality queen, you got to have good food, you got to have a good protective environment. I think that when I first started beekeeping and thinking about overwintering, I was thinking about all this stuff, maybe with the exception of varroa mites, all this stuff as something you were doing to help your bees in like December and January. But if we're really thinking about making those good winter bees, we really need to be thinking a bit earlier than that. And I have some ideas about how we may be able to do that, although I don't have any hard definitive answers. I just have some ideas. And I think that's something really critical. Part of the problem we have is that when a colony fails, it's very difficult to know what was the cause, or if it was a mixture of causes or whatever. And so what really got me interested in this and got me kind of to start digging a little deeper and kind of investigate this was starting, it's been probably almost 10 years ago when I talked to beekeepers here in Iowa, Nebraska, Minnesota, places, in the Midwest that were especially maybe a little colder, but not just colder. When you would have beekeepers lose colonies and you would talk about, well, why'd your colony die? Or when did it die? What happened? I think we often don't really know, or you think you did, you checked mites in August and then you didn't do it again, so you don't really know. But there were a lot of times where I think beekeepers would say, yeah, I checked mites and the mite levels were low and I treated anyway, and the colony had good honey stores and the cluster was, that big and it looked really good. And then it's just kind of petered out and died in like February or March. And there's still a lot of honey on and there's no sign of other diseases. And I don't know. And again, my gut reaction was always like, well, you probably had varroa and maybe you killed the varroa with like an oxalic acid treatment in December, but it was too late. Your colonies already had infections. They already had other stresses, blah, blah, blah. We don't know. It's hard to tell. Dead bees are hard to figure out what killed them in the middle of the winter, especially because By the time you actually get them out, they're pretty gross usually. But then again, this is something I saw myself where sometimes we would make a colony, and this was somewhat of an experiment, where you would take like 4 or 5 weak colonies and you combine them and make one really big colony, but all made of weak colonies. And they almost always die, in my experience. Very rarely would they succeed. Even though the clusters would appear really big and they would seem to do okay in the fall, they would eventually kind of peter out. That's just an observation. But it kind of made us have this hypothesis that maybe what we're seeing here is just that the winter bees that we're producing during that critical period in August, September, October, something's not happening to make them do really well and thrive. They're not able to make it all the way through the winter. They look fine. You look at the bee, and a bee is a bee, and it looks okay. And then those bees do well through December and January and February, but they just don't quite have it to be able to get all the way through. And one of the problems with testing this, well, there's quite a few of them. One is it's hard to sample bees reliably throughout a colony throughout the wintertime. And the other is before we started doing a lot of experiments, seeing if there were these problems, we really didn't know what the norm was, what a normal winter bees look like. And what do we expect it to look like under good conditions? And certainly people have been studying winter bees for a while, but not really in our landscape. And there are a lot kind of different factors that made it hard to compare. And so we started to ask this question, not just with winter, but the whole year round, and ask, how does bee physiology and disease susceptibility change over the season? So people had done work looking at disease presence in commercial beekeeping colonies throughout the entire year and kind of track them throughout the whole, people with migratory operations, or they move throughout the whole US, and people would sample bees from these colonies every 2 weeks for the entire year and measure virus levels in them, for example. And you would see that even in the same colony, every 2 weeks, they look totally different. And in some ways, I think people would be surprised by that, but I would ask you, if you were to just go to a random city of 60,000 people and just randomly grab 300 people and see what diseases they have in July, and then go back and do that exact same thing and see what diseases they have in December, would you expect to see exactly the same diseases? Probably not. There's probably differences in what diseases get transmitted, things like that. And so I think we see the same thing with bees, probably for different reasons, but the point still stands. So we knew that happened, but we didn't know anything about how bees changed and how they were susceptible. So we know from work that I've done and others have done that bee nutrition differs across the year. Bee nutrition affects how they survive against stresses, but we don't have good baselines. And so this is something that always bothered me, because people would say, well, okay, this is fine, but how does that look compared to a normal June or a normal July? So we started to look at our normal apiary and just started to record a lot more stuff about what the normal levels were. And in the apiary system that we have at the U of I, one thing that's important to remember is that we operate it in kind of a bizarre way from a beekeeping perspective, which is that, we aren't doing it from, we don't have to make a profit off of beekeeping, and it's not a hobby either, because it's like our full-time job. And so we spend a huge amount of resources to keep those colonies at like as much as we can top health, which means we do lots of varroa monitoring, we treat very regularly, we treat very properly, and we do a lot of feeding, and we just do a lot of stuff to try to keep the colonies very healthy. And then we do stuff to them that's maybe not so healthy, but we record that. And so we were really reasonably sure like this was good. It's a pretty kind of a good baseline. So how do you actually measure how healthy bees are? Well, there's a million ways to do it, and I've done so many of these. And so one way we've done a huge amount of this back when I was in Iowa is just going through and just exactingly measuring the populations of colonies, the weights of colonies, how much brood is in them, how much mites are in them, and taking pollen, you can see here's a pollen trap on a colony. So you can measure how much pollen is coming in and the identity of the pollen, the pathogen levels inside the bees. And there's all these different things that you can measure. And I think some are more useful than others. And so for this, what we started to do was just look at just general disease presence. Obviously, if a colony has a foul brood disease or something like that, we're going to record that, and that's going to be notable. We were keeping kind of careful track of mite loads using an alcohol wash method. We use those, I don't know if everybody's familiar with the Easy Check. It's like a plastic box thing or a little shaky cup thing. I shouldn't say it because it's like a branded item you can buy, but I actually don't really like them that much for a variety of reasons, but they are very replicatable because anybody can buy one and then they're all the same if you're doing an experiment. We weigh the colonies and then we can take samples of bees from inside them and we can weigh the bees. We can look at their fat stores, protein stores, and just stuff about the bees. But a lot of this is very passive, and people have done this a lot in both studies I've done and studies other people have done, where again you just grab bees out of a colony and you measure what are the disease levels they have. Again, you can track colonies as they move throughout the U.S. and just see what they have. But I was really interested in a more experimental approach. What happens if you just take a bee and you challenge it with a virus infection, a disease? How resilient is it to an infection? And so part of the reason we do this is because this is actually a method that I developed back when I worked at Iowa State, and we use quite a lot in my lab to evaluate the health and kind of the resilience of bees under different conditions. And so instead of just looking at the bees and trying to measure something, oh, they're heavier or lighter, it allows us to have a challenge and see how they react. And so basically what we do is produce, we figured out a way to produce basically pure virus particles of Israeli acute paralysis virus, IAPV. And basically we've developed this pretty nicely as a model virus system. So if you're familiar with bee viruses, if any of them, you're probably familiar with deformed wing virus, which is definitely a much more important virus from the perspective of bee diseases. And you'll see this in your colony. And IAPV is less important, I would argue, at least right now. But it makes a really great model system for studying and kind of testing bee health. And there's a couple of reasons. One is that almost all bee colonies I've ever measured, the vast majority of them already have some amount of deformed wing virus, but they don't all have Israeli acute paralysis virus. So you can kind of test that. And when they do have it, it's usually low. The other reason is that if you were to infect an adult bee with deformed wing virus, what happens? Nothing. They already have wings. The wings don't just like disappear. The wings are there. So they only get deformed if they get infected when they're developing. And so you can infect an adult and literally nothing happens. You have to kill them and measure the viruses in their body, and that's really hard. It's expensive. But with Israeli acute paralysis virus, if you feed them some or inject it into them, a lethal dose causes them to die they get paralyzed and die. And it happens very quickly within 3 to 4 days. And so you can do these experiments really quickly and really cleanly and kind of see how a resilience occurs. And we see this a lot of times where you'll have a treatment. Let's say you treat bees with a pesticide, for example, and nobody dies. If you put them in a cage, like in this picture here, feed them a pesticide, nobody dies, everybody's happy. But then you feed them or inject them with virus, and then the ones who are exposed to the pesticide die more or die less. And so you can kind of get at things very well that way. There's definitely an aspect of this that's when you own a hammer, everything looks like a nail. But this is a system that I really like and I think is really informative for telling us about how bees deal with diseases. And so like I said, we do this a heck of a lot. So we have whole rooms, we infect all these bees and we put them in little cages and we count how many bees die, measure how much virus levels are in them. And one thing we noticed doing hundreds and hundreds and hundreds of these is that while it's usually repeatable, so here's a survivorship curve. These are really boring, but it just shows you over time, from 0 to 72 hours. We feed the bees just sugar water, 100% of them basically survive. And then if we feed them, this is the most concentrated dose, it's called -2. It's just a order of magnitude dilution. It doesn't really matter. This is the most virus, second most, then less. We see that you give them more virus, more bees die, right? That makes sense. That looks really clean. And so on paper, it sounds really clean. And if you see me give a talk like this, I'll often show this and say, look how great this system is. It's so clean. But the reality is that when we actually look at survivorship of, let's say, the same dose, this -2 dose, which is the one I just showed here that caused the most mortality, and we just feed it to bees, every month of the year, the survivorship is a little bit different. And in some cases, like in July and August of this year, it was super different. But of course, these were experiments where we took day-old bees, like a frame of bees, and then pupae hatch and they emerge as one-day-old bees, and we feed them this virus. And you can't do that in the wintertime. You can only do that in the summer when there are bees being, eggs being laid and bees hatching. But this gave us this idea like, wow, there's so much variation. What happens during the wintertime? Are those bees in the winter being really, are they really weak? Are they really susceptible? And so we had this aim in our project to track mite levels, to track colony health the whole year for all the bee colonies in our apiary system, which is, I think, in the end, about 60 colonies that we tracked. We looked at the resilience of bees to virus infection in a subset of those colonies, and we tried to follow those same colonies the entirety of the year. And we did it by just sampling nurse bees. We went in, found a frame of brood, randomly selected a bunch of nurse bees, brought them back to the lab, and then we injected them with virus infections, saw what happened. And because we used nurse age bees, or what I should say, core of the colony bees, even in the winter, we could collect bees and get them from the middle of the colony and still test them. And the goal here was to be able to see, are there key periods in the year when bees are most at risk, when they're the most susceptible, when they're the most in need of beekeeper interventions? It's all fun to say, of course, you could feed bees more, treat bees more, but resources are finite. You can't do that all the time. So can we identify when is the most efficient, when is the most important time to make those interventions? And so we predicted that winter bees would be more susceptible to infection as the winter went on, and they would start to run low on resources, and we'd start to see them die more. And so just to start with, here's some of the data on our mite levels in our colony system. And so this was, again, from about 50 or 60 colonies. And again, this doesn't show in the winter because while we actually did take some mite samples during the winter, we didn't take the full 300 bee alcohol wash because we didn't want to take too many bees from the colonies, but we did during the normal season. And so this is the mite count per month. This is per 300 bees, which when I publish this in scientific journals, we always change it to percent, but I find for beekeeping audiences, having it in the 300 bees is more useful because you're used to actually looking up and seeing. And so here we can see, here's a colony that had a really high infestation comparatively, but those were pretty rare. And what we actually see is that for the majority of the year, our colonies had below a 1% threshold. On average, we were getting, sometimes less than 1 per 1 mite. And then even at our worst time of the year in October, we were getting 9 mites per wash, which is about 3 mites per 100 bees, which is still right at about a relatively kind of loose treatment threshold. And then once we hit that threshold, we actually did treat, and we treated using Amitraz, I believe, that year. And so we usually do treat. So I point this out because the results that we have, we actually don't think are due to mites. We control mites pretty vigorously, and I don't think mites are behind any of the results that we have, although we can't really rule it out, but I have reasons to think they're not. And it's important because we do know that high mite counts are really problematic overwintering. And people have done studies on this before and shown that obviously mites cause virus transmission, a lot of different viruses. Varroa mites not only transmit viruses, but they also feed directly on the fat body, which is this kind of super organ that insects have that is part of their immune system. It plays a role like their liver to detoxify chemicals. So it's how they detoxify pesticides they might be exposed to, but it's also their fat stores, just like, their extra nutrition. And so varroa mites bite onto the bee and they actually eat up some of that fat body. So what's it do? It's doing a lot of different stuff. So it's a really important organ. And we know that some previous work has shown that when, and this is a study where the black line is bees that had varroa and the dotted line are ones that didn't, that the bees who have varroa are less, they have less of vitellogenin, this protein they use to make royal jelly. And so we know that varroa parasitization does matter. So that's why we rule that out. We really don't think that that is playing a role here. And again, previous studies have shown that using just existing virus infections, not experimentally injecting bees with any viruses, that they sometimes do see this association with overwintering colonies having high virus levels, and that they're bad. That's bad. It's bad to have a lot of viruses in your wintering colonies. Some problems here. And so one of the really cool advances that we've had in the last couple of years of the UIUC Bee Lab is we got one of these overwintering buildings. Gosh, this is just so cool. At first, I thought it was a bit of a gimmick. I'm not going to lie. I thought it was a little gimmicky for us. These are buildings that, like I said, out west really started with people using them in like apple sheds and potato sheds out in like Idaho and Washington and Montana. And they've become pretty sophisticated lately. And they allow you to control the colony's environment really well during the wintertime. And we got one. And for me, it's cool, but it's less about really improving our own wintering success and more about creating an environment where we can keep bees in the winter and then still be able to do experiments on them and access them. Because the problem with studying winter bees is that if it's wintertime, your apiaries may be inaccessible due to snow or mud. If the weather's really bad, you cannot open the colonies without stressing them a lot. The bees all fly out and then much of them die. That's a problem. And so this building allowed us to track colonies really well. And so we basically had these set up to be about 40 to 45 degrees the entire winter season long. So it's cold in there. And it's got airflow and it's kept completely dark. And so that's a temperature that's been shown to be metabolically the most efficient for bees. So it's just hot or warm enough to where they don't have to eat tons of honey to keep themselves warm, but it's cool enough where they stay in a cluster. It's basically like almost the coldest they'll be to reliably really stay in the cluster. So it's really nice. And so, but that means that we could have our focal colonies here and go into those colonies and collect bees out of them safely in the dark. We have a red light headlamp on and you collect your bees. This is just a list, I shouldn't. So then we would bring them back into the lab. This is Gyan. In my lab, we go use a little weird little bee vacuum, suck them all up into this little vacuum. And then this is carbon dioxide. We knock them out with carbon dioxide. They just they fall asleep, they get anesthetized for a couple minutes. And then here's Gyan injecting a little bit of this virus inoculum into the bees. And so this way we're able to test throughout the entire year, summer and winter, how susceptible bees are to different types of infection. And so this is like a crazy looking graph. And the pattern here is the main thing you're looking at. So this is back in February 2021, and there's some more months that I'm not showing here. And it just goes February, March, etc., all the way down to January of 2022. And here on the bottom axis, we see that black bar is bees that didn't get injected with anything. Gray is ones who just got injected with basically water. And then we're going with a virus injection that goes from very low amount of virus, 10 to the minus 14. So that's like 1 quadrillionth as much as what the most concentrated we could do. And then the red is the most concentrated. And so we can see if we just focus here in like our field season, like May, we can see that if you just, put these bees in a cage, very few die, like less than 10%. And then to kill any bees with virus, you've got to inject them with one of these 3 doses, these 3 highest doses. And that's the case in May. June's a little bit different, but not too different, a little bit more resilient. In July, our bees are tough as hell too. They have they require these 2 highest doses of viruses to die. And then it changes dramatically. And this fits what our observations have been for years, that in August and then in September, the bees become extremely delicate. They are very susceptible to infections. Even really low concentration amounts of virus kills all the bees. September comes, same deal. And then this is where we started to get really surprised. They started to become more resilient again. And so in October, it's in the middle, but then November, December, January, and I don't show this, but we even go a little bit further than this. And we see that these bees are actually really resilient through December and January. And again, if we just look at this in a different way, this is just looking at bees over time. This is if they weren't injected with anything. This is if they were injected with a little bit of virus. This is if they were injected with a lot of virus. And we can see that if you're injected with a lot of virus, you're probably going to die. But if you're injected with a little bit of virus, the amount of bees that die is at its highest in April and May, and then August, September, and October. But then the bees are really resilient in like January and February and December, which really surprised me. When we look at the fat bodies in these bees, so how fat are the bees, it actually kind of makes sense, but doesn't fit exactly perfect. We actually see our bees are at their fattest in February and March, which is a bit puzzling because they're the same bees, right? The queen has basically stopped laying eggs here. And so these are the bees that are just kind of in the colony, getting along, and they're getting fatter. I don't think so. I don't think they're actually getting fatter. If we think back to that figure I showed earlier where you have a bunch of winter bees in the colony, but there's also some summer bees left, I think what's happening is throughout this time period here, the last of our summer bees are dying. There's still some in there and eventually they're dead. By the time it's February and March, the vast majority of them are dead. So when we go in and just randomly scoop out some bees, we might be getting 75 or 75% winter bees here. But by the time it's February or March, it's like 95, 100% winter bees. So what's this all mean? Well, I really think that shows that winter bees can be really tough under the right circumstances. And this makes sense. Honeybees have to get through this really tough time of year. Winter, there's no food, they got to stay in the colony. And so to do it, the bees that got to do it have got to be tough. They've got to be resilient. But they also don't have a lot to do. They don't, if we think about the resources their body has to use, they don't have to care for brood. So they don't have to make a lot of royal jelly. They're just like feeding the queen a little bit. They don't have to forage. They don't have to go collect food. Hopefully there's enough food in the colony for them to eat so they can eat nectar or eat the honey and they can have some pollen. And so if there's a disease, they can invest everything they've got into fighting that disease. And they better do it because there's no second chances if the colony dies, right? The queen can't make more eggs or make more bees in the middle of the winter time. And so in the right conditions, these bees remain tough for a really long time. And then other studies have shown that once those winter bees smell brood, within about 3 days, they start to change and they start to just become normal nurse bees. And so they really are just holding on until that happens. We also saw that summer bees fluctuate a lot. Summer bees have to vary a lot with the season. Temperatures change, humidities change, the weather changes, food is really different, diseases are really different. And we see that during sometimes the years, they're super susceptible to stress. Again, we use viruses to test this, but I kind of think that this isn't special. These pathogens aren't special. We've only seen this a little bit, but I suspect that bees are more susceptible to other stresses too, like pesticide exposure, for example. We don't really know that for sure, though. So how would you translate this into management recommendations? In some ways, I think it's a little early, and it's hard for me to be 100% certain. So take this with a grain of salt. But these are the thoughts I have. So these are more like hypotheses that need to be tested. And I'm curious what people think. First off, monitoring and treating for mites is, I think, really important. This is a whole separate issue. We did this big survey, beekeepers in Illinois, and we find that a surprisingly large number of beekeepers don't monitor for mites or treat for mites. And I actually understand. I feel like I understand reasonably why. But if you have really high mite levels, I think that's going to throw a wrench into everything. High mite levels are going to make everything worse. I think another recommendation is just know your area. Know when food is coming in. Know when your bees are bringing in lots of pollen or a little bit of pollen. This can be harder than you think, because when you see pollen coming in, it's very hard to know what kind it is. And we know that different pollens help the bees in different ways. And so that can be really tough. In my opinion, even in Southern Illinois, it's probably a little bit different, but I suspect in that August to October range is still when you're going to see the biggest trouble. It's usually post clover bloom. Going out on a limb, this is like contravenes a lot of management instruction, but I like to feed pollen patties, just like the regular protein ones, in August. Basically, they're on the time that we take off honey supers or do a mite treatment. This is something I learned to do in Iowa as well, which was atypical there. And it's become more problematic with small hive beetle pressure. Wheat colonies, I know it's a problem down in Southern Illinois, and it gets worse every year up here. And I don't think it'll be long before it's even worse further up into the state. One thing, this is just something we do sometimes, if you're not sure if a colony is getting proper pollen nutrition, you just give it a little piece of a pollen patty and you see if they eat it. And if they don't eat it, then that often means that they are bringing in enough of their own pollen. If they eat it really fast, then that means they might not be. And then I think a big problem that we see is coming out into the spring. So your bees are going to start to they're going to lose that winter bee tough guy ability as soon as they smell brood. And again, people have studied this before, that it's that switch of smelling brood, then they got to take care of the brood, and they can't put all their resources to fighting disease anymore. They got to give the brood some food. And so that late winter, early spring is such a dangerous time. And I think that's when we see our biggest problems, is times when you have a really early thaw and food becomes available and the colony starts to raise brood, and then it gets cold again and the food shuts off, but the bees can't go back. They can't go back to being winter bees. And so they've kind of gone down this path of no return. And if it remains cold too long, it becomes really hard for them. And it becomes really easy for them to starve or just become very stress intolerant. But yeah, like I said, good winter bees can take a lot of stress. They're really tough. But we've got to really work to set them up for success, both in the fall and in the spring. And then, like I said, some of the stuff we've been doing in my lab has been to look at how maybe we can buffer them against some of the fluctuations in the winter, either with hive wrapping, or I'm becoming increasingly impressed by the ability to buffer them from changes inside of a building. Although there's kind of a big expense to that. I'm curious if it will ever become scalable. So that's all I have. I mean, and I've taken up the majority of the time. And so I really am curious, what do people think? What are you guys seeing? When do you see brood rearing ending? When are you seeing pollen stopping coming in? When are you seeing when you have colony losses, when do you notice them? Yeah, people can type questions in the chat, or you can just say them. It's up to you, whatever you prefer. I'll just ask everybody a question. How are has anybody had problems with small hive beetles this year? And is it worse than any previous years?
Yeah, I think they've been worse this year. Considerably worse for me.
Yeah, Same for us. We were trying to do an experiment actually this year with about 20 nucleus colonies, which, is always a little risky in the fall. But I was just shocked. They just got blasted by hive beetles. Within a week, they were toast. I mean, they just got filled with beetles, and they got slimed pretty hard. It was crazy. I never seen anything like it that bad. Yeah, but yeah, I'm curious if it's this year in particular with beetles, and if it'll be better next year. But I don't know. I'm very interested in beetles, in small hive beetles. So I think, it's something you often see. Strong colonies can take care of it, but weak colonies are when you really see it. And we often set up weak colonies kind of on purpose for experiments. And so we see it a lot, but in the past we just haven't. This year has just been extra bad. And I just had a visitor, a friend of mine who works at the USDA's bee breeding and genetics lab in Baton Rouge. And she said they can't do any mating nukes or anything like that because of beetles. So yeah, someone asked in the chat, what's the preferred method of treatment for hive beetles? This is tough because small hive beetle, historically we use traps, those little oil trap, the little where they fall in. I don't know if they even work that well. I think that they work okay. I mean, beetles fall in them, but I'm not convinced that they maybe help a strong colony keep it under wraps. I think keeping colonies strong is the biggest thing. We actually are working on a project now to try to find some like novel kind of pesticide type product that would be able to kill beetles without harming bees, but we'll see. Nematodes is an interesting idea. People have been used diatomaceous earth or things like that in the soil. But yeah, it's different. So how will small hive beetle affect virus susceptibility during the winter? I've seen small hive beetle overwinter with the bees. Yeah, so we see them overwinter in the bees too. And that's one of the things I'm really interested in, because most of the beetle work has been done in the South or in other countries that are warm. And so the focus is always on beetles overwintering in the soil, because they come out and they go in the soil and they pupate there and they just hibernate, and then they come out in the spring. But ours don't do that. At least up here, it freezes enough that they all would die. So they're in the winter clusters. The adults are. And so what are they doing? There's almost no research on what they're doing in there. And we actually have a project ongoing because to test the virus, some virus stuff. Like, do the beetles harbor viruses? Do they spread viruses around? I think our evidence actually shows, luckily, they don't seem to carry any of the viruses, and they don't carry them for very long if they do. I don't think it probably harms susceptibility, but what I do think happens is that during that time of August, when we see the bees that they're most susceptible to viruses, it's also, unfortunately, the time when they are more likely to encounter high mite levels and small hive beetles. And so I think that that's where you can just really get in trouble really very quickly. If you have a colony that's looking strong, again, we don't know what it is, and then something happens and then mite levels go up, and then they become more susceptible to infections, then a lot of the bees start to die, and then small hive beetles move in, and then your colony can just collapse very quickly. Is it normal in October to have little to no eggs, larvae brood, lots of bees, and lots of honey resources? I would say that would be normal here if it had been kind of cold. If you have some eggs and larvae, depending on the queen and depending on the queen breed. Some are going to push longer into the season than others. But if your queen is present, and you have some eggs and larvae, and there's strong bee population, then I would suspect that colony is kind of doing what it's supposed to be doing. Does anybody feed pollen patties in the fall? I'm always curious about that. almost nobody does. I'm very torn about it. I learned it from a beekeeper in Iowa that I had worked with who was just very successful at overwintering there, thousands of colonies. But it makes me nervous because of our problems with beetles, for sure. Yeah, it makes it, yeah. Yep, there's still some stuff out there. Goldenrod's still blooming a little bit up here, so I suspect you guys have it down there still. Asomasters as well. Yeah, somebody asked in the south, do the bees go through the same winter bee cycle, or are they all summer bees all year? Yeah, so winter bees in other climates do go through a winter bee like phase. And there was actually a study came out a year or two ago in Puerto Rico, so tropical, and they do go into a winter bee phase, and it's just not cold. It's just when there's no flowers blooming. And so that's why calling them winter bees is in some ways a misnomer. They're really just like stress tolerance bees, because no matter where you live, that's not totally true, but many places, even if it doesn't ever get cold, there's usually a period of time where it's really dry or it's really hot, and plants just stop producing nectar and pollen. And so the bees have to do something. And so like in these tropical areas in like Puerto Rico, they have a dry season and or a really rainy season, and then they don't do that. They can't forage, and so they do that. But it can be shorter or longer depending on where they live. And yeah, my friend down in Louisiana is actually hoping to replicate some of this work down in Louisiana and see how things are maybe the same or different. Let's see. Yeah, I mean, we usually don't have tons of problems with pollen being coming in or not, but I don't know. It's just cheap insurance so much. Quit the high fat content bees in February, March. We do with early pollen collection. Our bees collect a lot in those months. That is possible. It's very hard to tell that question. So, because one of the hard parts with, bees collecting pollen is you see bees collecting pollen, but are they eating it? And if they are, who is eating it? So that's weird. In those examples, though, in our experiment, our bees were still inside of our building, and so they were not collecting pollen during that time period. Although we also do have pollen collection even in February because of willow blooming. Yeah, Don, you said that if they take dry sub, they need wet sub. I don't use dry sub ever, so I haven't really used that much. St. Louis to West County. Yeah, that's interesting. The heat issue in urban stuff. Yeah, there's some work on that. Some actually kind of interesting work on the Heat Island stuff out from like Raleigh, North Carolina, where they had done a lot of stuff in the city and then out of the city, but I can't remember how much of an effect they saw. Yeah, Charity, you asked if there were tubes in the building for ventilation. No, actually the tubes are really cool. So because we want everything to be multi-purpose in our building, so we can put it this is our wintering building, but during the summer, we can actually do other stuff. And so these tubes are ports so that we can hook up beehives, either observation hives or other manipulatable hives, and have them flying out into just open forage, but then have the colonies inside. It's primarily for observation hive experiments where we want to be able to observe what's going on. My lab actually doesn't do that very much, but we share the bee lab with another lab, so they do. How do bees and beehives eventually get rid of a virus infection if they don't have immunity memory like other animals? Does it keep reinfecting? Does it cycle through every bee? Is it dangerous to use frames from a die out in another hive? That is an awesome question. So they do have a little bit of immune memory. It just doesn't work like ours, right? They don't have antibodies, but they do have antiviral immunity that does have a short memory. And there's even some evidence for some pathogens, although it's ambiguous, it works for viruses, where if the queen gets infected with or exposed to a pathogen, she'll actually, when she makes an egg, put like a little chopped up piece of the pathogen in the egg. And so when the egg hatches, it elicits an immune response against that pathogen piece. And so it's not like it's not an antibody, but it kind of works like that. But your question about reinfecting is actually really interesting, and it's something we're working on right now because we have no idea. Again, with deformed wing virus, which is what most people work on, it doesn't matter. If a bee gets deformed wing virus when it's developing, it's got deformed wings and it's pretty screwed up because it's developed wrong. And so whether it gets reinfected or not, no one really cares. But for Israeli Q paralysis virus, we actually have had that same question. How long does it last, whatever. And we've actually found that if a bee gets infected with IAPV, she either, she dies or she doesn't. And if she doesn't die, she usually has completely cleared the infection within about a week. And it appears normal. And we haven't done full lifespan studies, but she seems to live for a reasonably normal life. But we don't know about reinfection. We're actually going to try that next year. And I suspect there's got to be some cost to it. Is it dangerous to use hives from a die-out in another hive? They almost always say yes. And I think it's likely to some extent true. So while the bees, when they can clear an infection really quick, some of these viruses are really persistent in the environment. And it kind of makes sense because before Varroa, how did these viruses get spread? They didn't have Varroa to move them around, but they still existed. They almost certainly got moved on flowers. And that's why they weren't that big of a deal. But how do you, to survive on a flower, you're in the full sunlight and it's outside and it's hot. So they're pretty tough. And so we've done some experiments and we find that if you just make a little tube up of IPV virus and you put it in a incubator at 95 degrees Fahrenheit, that virus will survive very well in there and kill bees for 3 weeks, more and infect them for probably longer. And so the frames from a die-out can certainly have probably live virus particles on there. Is it really dangerous to do? To be honest, I have never actually seen any evidence that it's meaningfully dangerous to a colony for viruses. Obviously, American foulbrood is a very different story. Anything like that. But I would be very surprised if it would matter, because when we feed bees virus particles and we infect them by feeding it to them, you have to feed them a really large amount of virus to get them infected and die. And so I suspect that on the frames, it would probably not be a big amount, but if you read recommendations, they almost always say don't do it. That said, I have reused many frames from colonies that have died of mega mite infestations with no negative consequences. So I know that there are some places now where people put them in ozone chambers and it can kill all the pathogens, but obviously I don't have one of those here. But one thing people I have done is put them out in the sun, or spray them down with isopropanol or something that's going to evaporate. But does it matter? I don't know. So when do they stop storing? What indicator that they will need feeding through the winter? So this way, gosh, when do they stop storing? So when will they stop storing? That's a good question. I mean, I think a lot of bees will store as long as they have room and you keep feeding them. But if they're collecting pollen or nectar, some of that comes down to the genetics of the bees. Let's say, is that the paper I've? Oh, that's like a mislabel one. I hadn't seen that, Don. I'll have to take a look at that paper. Thanks for sending that. But yeah, like the best way to feed, that's a good question. I don't know. I bet everybody has an opinion. We have historically always fed heavy syrup in the fall, and I have used both corn syrup as well as like heavy sucrose solution syrup. And both have worked fine. I know some people don't like corn syrup. And then I know a lot of people that like the dry white sugar, either like white sugar cakes, or the bee inspector for Iowa used to just pour white sugar on a piece of newspaper. I don't think the bees like that a lot, but they'll eat it if they have to. Same with buying those, like they make those like winter patties. And I've used those before, but I never really feel like the bees eat them very much. And maybe that's a good sign. Maybe that means they had enough other food and that it didn't get into the emergency food. So I didn't talk about it much, but when we did this wrapping study, we fed them sugar syrup and also they had honey, and then we gave them hard like fondant patty, like homemade fondant, just sugar patties. And we measured how much of that they consumed, and we did see that wrapped colonies ate less. So I think that that is nice. If you have moisture control problems, I think that the white sugar ones, the dry white sugar ones, can be really nice because they help absorb moisture. So they have kind of a dual role. They're also really easy. And like I tell everybody I work with, I'm really lazy. And so if something's easy and it works, I usually am in favor of it. I think most people would argue honey is the best. But I think one thing that's often hard to evaluate that people sometimes forget is how much pollen is stored. You do want to make sure that you have, if you can, several good frames of pollen in there, so they have something when they come out in the spring. The other reason I like those dry patties is that you can add them midwinter if you have to. And so it depends. Yeah, like you would ask somebody, you'd ask what indicator that they will need feeding through the winter. So there's different ways to kind of weigh them. And I think a lot of people just get a feeling for how heavy they are. And so if you feel like a colony is getting kind of light, or, I should actually just post this on our website, but we use this very simple scale to weigh colonies during the winter. And it's basically, you just go on like Amazon and buy, it's called a crane scale. And they're usually marketed for weighing animal carcasses, like for deer hunting and stuff like that. It's like a little box, and it's got a metal, you can put a metal shackle or like a big carabiner on it, and then some kind of any kind of hook on the bottom, and then you kind of hook it under the hive and you lift it up and it tells you the weight. You do that on both sides, and that can give you a weight estimate. And so then you can think, okay, my colonies ate 20 pounds of honey, 30 pounds of honey, 50 pounds of honey. Maybe I need to give it some food, but you don't need to open the colony at all to do it. So we do that sometimes as well. But we also, though, definitely overfeed. We definitely end up overfeeding rather than underfeeding in almost all cases. So is stored pollen in the frame better than feeding pollen sub in early spring? I wonder how well the protein value is maintained over the winter. I generally think, yeah, pollen is usually better than pollen supplement. The hard part is that every pollen is different, and some pollens probably are worse than pollen substitutes, and most are probably better. There is evidence that the protein value declines over time. However, there are some other changes that maybe make it good, in terms of like what nutrients are made more available to the bees that are in stored pollen. again, if you give them a choice, they will usually consume real pollen. Again, when people have done that, I usually err on they give them some real pollen. In my experience, the risk of small hive beetles is less in the spring. And so your problems aren't there. Like I said, if you're going in your colonies a lot and don't have a lot of them and it's easy to check, I often just throw a small piece, like, just like maybe 2-3 inches by 3 inches on top, and just see if they're eating it. And if they're eating that pollen patty within a day, 2 days, that means they want some more. But if they're really not, then I don't worry about it. And it's kind of a nice way to let the bees tell you if they need more. But obviously, if you've got 500 colonies, you're not going to do that. Although you could do it to like a couple, and see if kind of on the average if that's happening. But yeah, to me, it's very hard to tell. You see that stored pollen and it all looks the same to me, but it likely is pretty different to the bees. And then I will also add, this is another thing that we poorly understand, is if you did have a pesticide exposure event a long time earlier, that pollen gets stored, you don't really know what's in it, but usually your pollen sub is probably free of pesticides, I hope. I generally don't think that's a big deal, but it could be in some areas. I don't think it would be a big deal in most of our area here, though. But we use pollen sub a lot in our experiments for this exact reason, because when we feed bees, like in those cage experiments, it's really hard to know, you got some pollen here, and I want to repeat this experiment next week, and I run out of pollen. I can't just go find random pollen. Pollen is different. And so we use Mega Bee a lot because it's very repeatable. We can just go buy more Mega Bee, and it's theoretically mostly the same. And so bees do reasonably well on it. We kind of consider it as like, it's okay. Oh yeah, this might be a good time to plug the ISBA fall meeting. That's true. I think it's November 6th in Springfield. I'll probably talk about this again or in greater detail at that. Sorry. Yeah, so somebody asked, are there supplements that have been studied that provide a benefit? There have been some, yeah, there have been several studies that have shown benefits of some. I'm trying to remember with Honeybee Healthy if there have or not. I'm trying to remember, because some of them are weird because they have an effect of just being a feed stimulant, right? Like Honeybee Healthy, I think it may be that it just makes them eat more, which that's fine. Yeah, I can't remember with Honeybee Healthy. I haven't followed some of that antimicrobial stuff. Diana asked concerning insulation behind the sleeves. Can you purchase? Are there any other ideas to keep hives warm? Yeah, let me show you actually kind of a funny picture. So if you read the old books, boy, they got ideas for keeping stuff warm. So this is a Dadant method of wintering from 1923. If you can still see my slide. So Dadant had this method in the old 1920s book, which was you would take chicken wire and you put it around the column of the hive, and then you stuff it with leaves. And there's several different versions. So, and then here's this other one where people build like a wooden case. The moral of the story is there's so many ways that people have used to wrap hives in foam insulation or just buying like the pink board. And actually, again, I didn't really talk about our study with overwintering stuff, but one of the things I always joke is like, it's like in Ecclesiastes, there's nothing new under the sun. So like here's a book from 1968 when Albert J. Cox was doing coroplast sleeves, and people use those now, and then wrapping or putting them in buildings. This is a bee building from 1923. They're just a little bit more sophisticated now. You could use anything. The reason that we got kind of interested in it is that people would say wrapping works, but no one could explain what it was doing, and they would often contradict. So some people would say, oh, I use insulation, like fiberglass insulation keeps it warm. The heat from the hive gets kept inside. Okay. Then other people will use tar paper or what's it called? Sisselcraft paper, which is like black tar paper. And they say, oh yeah, that doesn't really insulate, but it's black. And so it absorbs the heat from the sun and it warms the colony. And it's like, well, those are two completely different things. Those do very different things. So what actually is it doing? And so we tested these, like they call them cardboard packs or coroplast sleeve style ones. But they also make these other style ones where they actually have like an insulation style in them. And we did this because we had beekeepers who were really interested in it and like helped us get them. And we actually found they are very effective. And I think it's primarily because they are creating just a little air gap that does provide insulation, but then they're also like a windbreak. It's like a little windbreak. So roofing felt, whatever, anything. There's also arguments for some European traditional hives. Tom Seeley makes this argument, like Polish traditional hives, the wood is like 2 inches thick. And yeah, that's probably great, but I'm sure it's heavy as hell to lift and carry around. So you can really wrap it in anything. I think the biggest mistake people make in wrapping is not ventilation, is not ventilating properly, or doing something where the colony traps moisture in a way that the moisture can build up and then drip on the bees. That is like a death sentence once they get wet like that. So I like the black plastic ones mostly for human reasons. They are relatively cheap, they're really durable, they're really light, and they fold up flat, and you can put them in your attic or up on the eaves in your shed or whatever when you're not using them. But yeah, roofing out, you can use literally anything. I think even just like a windbreak is valuable. Alex, you said unrelated wintering, but related resiliency, aviars in 5 acres, farmlands, no spray. So yeah, so do you research on the impacts of resiliency to spray exposures? Yeah, so one of those students in my lab does work on the interaction of some of this virus infection stuff and nutrition and pesticide exposure. And actually some of the stuff that we find is actually really weird and maybe surprising. So one of the things he found is that, so in that project, we use pesticides that we get the levels and the identities from a field study we were running in Iowa, where we actually had bees in corn and soy systems, and we measured what we're really getting exposed to, like in real life. And then we took those levels and we did experiments in the lab. And what we found was that with some of those pesticides, if you feed them to bees and then give them a virus infection, the pesticide actually makes them survive better than if they didn't have the pesticide, which seems crazy, but there's actually evidence for this in a lot of animals. And it's like, because the pesticide levels are actually quite low, it's like just a little bit of poison stimulates your stress response and makes you resilient to other stresses. There's probably a cost, but, and so we do work on that. And actually, yeah, so it's completely bonkers. We were very surprised. But then we also find that there's not just one thing that makes a bee healthy, right? So getting exposed to a virus, that's just one thing. So if we take that exact same pesticide treatment, which is usually mixed with pollen, it's a mixture of insecticide and 2 fungicides. We take that and we feed it to a colony, like a micro colony where they're feeding a queen, nobody will die. The pesticide level is really low, but the queen will lay significantly fewer eggs. The workers will take care of the queen more poorly. And then there's a host of other kind of queen related challenges. And so, yeah, maybe the workers aren't dying of infections more, but they are responding to the queen differently, and the queen's health is compromised in some way. And so there's lots of different ways to be a bee. And I actually think we don't have strong data on this, but I suspect that over time, just like we saw that August susceptibility to virus, I suspect we see the same thing happen with pesticides, at least for some pesticides, that they're just susceptible to stress sometimes of year. Like we just sometimes have like so I share the B lab with another research lab group. And in this August time, we were doing all our experiments and they were setting up all these big experiments and they were freaking out because all their bees were dying like all the time. They were just marking them and putting them in an observation hive and everybody was dying and they were like freaking out. And I was like, yeah, it's August. That's kind of what happens, unfortunately. And it wasn't anything weird. They weren't giving them pesticides or viruses. They were just kind of handling them a lot. And I think that made them all get really stressed out and died. So I think that it would likely spray effects and pesticide exposure, that almost certainly has a similar response. If a hive goes in a winter without all the frames full, with the unfilled space affect the temperature in the hive? Well, it depends how full. Like if it's just air, I think that's probably harder for them to thermoregulate a little bit because there's less insulation in there. But if it's just like drawn comb with no honey in it, I don't think that's going to be a problem. The bigger problem I think you might have is sometimes bees do weird stuff in the cluster, and the cluster like goes in I'm going to say like a dumb place, and they just move somewhere bad, and then it gets really cold, and then they can't move, and then they all die. I've seen that happen multiple times. And so you kind of want to constrict them, but people overwinter colonies in nukes with just 5 frames sometimes, so they can certainly survive in those smaller settings. But do you have to have like every frame totally full? I don't think you have to. And certainly people use 8-frame equipment or overwinter in smaller equipment, so that's certainly possible. When I first started it here at the UI, our B-Lab manager at the time, they wrapped everything in it's like that shiny silver stuff that you put in your windshield, like the sun blocker stuff with like the little circles. And I still was puzzled by that because it was like, that's reflecting the light. Don't you want it to absorb it? It had some insulation property, but I hated those things. Well, thanks everybody. I appreciate it. It was a good discussion.
Yeah, thank you, Adam. We really appreciate the presentation, and I found it very informative. So thanks again.
Sure. Well, yeah, like I said, it's my pleasure. And yeah, maybe I'll see people if they go to the state meeting, or I'll see people down in the area sometime. So.
Sounds good. Thanks, Adam.
All right. Well, have a good evening, everybody.
You too.